Experiment 2, Analysis of Carbohydrates Part. Osazone Test. Introduction. The technique was developed by Emil Fischer, German chemist. Osazones are formed when the sugars react with a compound known as phenylhydrazine at boiling point. These sugars are reducing ones which have either a free aldehyde or ketone group to react with the phenylhydrazine. Materials required: the test solution, phenylhydrazine hydrochloride, glacial acetic acid. The procedure: weigh out 1 gram of each sugar provided into boiling tubes separately and dissolve it in 10 milliliters of distilled water. Add 2.5 milliliters of phenylhydrazine to each test tube, then add 3 milliliters of anhydrous glacial acetic acid to each test tube and mix well. Keep in a boiling water bath for 20 minutes. Note the formation of yellow crystals from some carbohydrates. Observe crystals under a microscope. Results and discussions. The reaction is stepwise. Firstly, the carbonyl group reacts with phenylhydrazine giving phenylhydrazone by elimination of water molecule from the functional group. In the next step, 1 phenylhydrazone reacts with 2 phenylhydrazines. The resultant product is osazone. Osazone formation involves hydrazone formation as C1 of an aldose, or C2 of a ketose, and oxidation of C2, or C1, of an alcohol group to a ketone, or an aldehyde. The new carbonyl group is also converted to a hydrazone. Osazone crystals have a characteristic shape under the light microscope and help in the identification of the sugar type. Making osazone crystals. The difference in the structure of the monosaccharides is caused by the diverse groups attached to the first and second carbons of the sugar molecules. Their needle-shaped crystals show that the position of the first and second carbons do not matter in the crystal formation. Test for galactose, mucic acid test. Introduction. The mucic acid test is used to identify the presence of the sugar galactose. When galactose solution is heated with concentrated nitric acid, a dicarboxylic acid called mucic acid or galacteric acid is formed as a white precipitate, which counts as a positive result. Materials required: the test solution, concentrated nitric acid. Procedure: treat 1 milliliter of carbohydrate solution with 2 milliliters of concentrated nitric acid. Let the test tube stand in a boiling water until effervescence commence and red fumes are evolved. Now transfer the test tube to a water bath maintained at 65 to 70 degrees of centigrades. Allow to stand for 15 minutes. A white sandy precipitate, mucic acid, indicates the presence of galactose. Results. Discussions. The aldehyde and primary alcohol groups are oxidized to carboxyl groups in the reaction of galactose with HNO3 forming saccharic acid. The saccharic acid formed from the free or combined glucose is insoluble and separates out as colorless crystals in the cold. Application. Galactose is a component of the antigens present on blood cells that determine blood type within the ABO blood group system. Determining galactose present on urine. Galactosemia, a genetic in which an enzyme needed to metabolize galactose is deficient or absent, typically develops shortly after birth. A galactosemia test is a blood test, from the heel of the infant, or urine test that checks for 3 enzymes that are needed to change galactose sugar that is found in milk and milk products into glucose, a sugar that your body uses for energy. A person with galactosemia doesn't have one of these enzymes. This causes high levels of galactose in the blood or urine. That is the end of the second practical. Thank you.